This lesson's on translocation in the phloem. Translocation is the term given to the movement of the products of photosynthesis, the sugars made in photosynthesis, and translocation happens in the phloem. The phloem is another vascular tissue. We've looked at the xylem in a previous lesson. Now, the main solute in phloem is sucrose, a disaccharide made of glucose and fructose, and that's what's transported in the phloem from one part of the plant to another. Now, the phloem is made up of two types of cells: sieve tube cells, which are cells that are joined end to end, and that at maturity have perforations in the cell walls between them to allow the sap to flow more easily. And another kind of cell called companion cells, and the companion cells are organized adjacent to the sieve tube cells. So, for example, these might be the companion cells, and I'll draw the nuclei of the companion cells in here. Now, the sieve tube cells, these ones over here, are alive at maturity. Now, that's different from what we saw in the xylem, where in the xylem cells are actually dead at maturity and completely hollowed out. The sieve tube cells are alive at maturity, and they're not completely hollowed out, but they lack nuclei. They've lost their nuclei, and they also don't have many organelles. They don't have ribosomes, etc. And that's in order to allow the sap to flow more freely. Now, because they don't have nuclei or other organelles like ribosomes to make proteins, they can't support themselves, and therefore they need to be supported by the companion cells. The companion cells support the sieve tube cells, performing many of the functions that a living cell would need, and for example, passing proteins, etc. into the sieve tube cells to keep them alive. So that's the structure of the phloem. The function of the phloem is to transport sugar, as well as other substances like amino acids, for example, from sources, places where they originate, to sinks, places where they're going. Now, sources include things like sites of photosynthesis. Mature leaves that are photosynthesizing are making glucose, and therefore they are a source of the sugar that's going to be transported in the phloem. And a sink could be a growing part of the plant, and that could be immature leaves, or it could be growing roots, etc. So the phloem is taking sugars from sources to sinks. Sources can also be things like storage sites. Tubers like potatoes, for example, are places where plants can store sugars in the form of starch over the winter and then use that in the next season, in the next growing season, to grow. So sources can be sites of photosynthesis or storage sites. Sinks are basically growing parts. It could be growing leaves, growing stems, growing fruits, etc. Now, sources and sinks can change. For example, an immature leaf would be a sink. It would need sugar to build cellulose as it's growing, but when it matures, it becomes a source. And so sources and sinks can change over time, and therefore the direction of translocation can change as well. So translocation in the phloem is not unidirectional, and that's different from what we saw in the xylem, where water was transported from the roots up to the leaves in one direction. Let's look at the process of translocation in a little bit more detail. Let's draw some phloem here, and we'll imagine that our source is a mature leaf and our sink is a growing root. So first, sugar is loaded into the phloem at the source by active transport, and that's because the concentration of sugar in the phloem is already higher than that in the source. So we need to use active transport to move the sugar against its concentration gradient. The movement of sugar into the phloem creates a concentration gradient that pulls water in by osmosis. So now there is sugary water in here. This is the phloem sap, and that creates a pressure gradient because we've got more fluid in the tube over here. As a result, the whole thing, all the sap, will move towards the sink by bulk flow. Remember, bulk flow is the flow of a fluid in response to a pressure gradient. So again, just like we saw in the xylem, we've got movement by bulk flow, but here it's different. In the xylem, the transpiration was creating a very negative pressure that was sucking the fluid along by bulk flow. Here we're creating a positive pressure by moving sugar in by active transport, and then water's following by osmosis. So the sugar and the water move to the sink, and then at the sink, the sugar is unloaded by diffusion, and that's because the concentration of sugar in the sink is lower than that in the phloem, because the sugar is being used for energy, or it's being made into cellulose, for example. And as a result, the water moves out again by osmosis. So sugar is unloaded from the phloem by diffusion. And that water might be moving into the sink, or it might be getting recycled into the xylem, which is adjacent to the phloem. But either way, both the sugar and the water are leaving. So that's a look at translocation in the phloem.